This is protein structure 1. You can see the vocabulary list here as well as the learning objectives. So the topic for today is about protein structure, and the key building block of proteins is amino acids. Before we get into that too much, though, I just want to give a little introduction about why we're talking about proteins so much in this course, Molecular Biology of the Cell. So recall that the central dogma of molecular biology is that DNA is used as a template to produce RNA through the process of transcription, and RNA is a template to produce protein through the process of translation. In other courses, such as genetics, our 312 class, you'll hear a lot about DNA and RNA as the focus is on genes. In this class, Biology 313, our focus is on proteins instead. And so together these courses can fulfill excellent view of looking at core tenets of molecular biology which underlie all cells and organisms. So to talk some more about amino acids, recall that amino acids, or as I will abbreviate them, AAs. Amino acids, these are the building blocks for proteins. So amino acids are also known as monomer. Mono meaning one and mer meaning unit. And amino acids, when they are bonded together in a long chain, produce a polymer called a protein, or polypeptide. We'll talk a little more about the specifics of that vocabulary later. For now, you can use them interchangeably. And again, this is a polymer of amino acids, AAs, that are bonded together covalently in a repeated pattern So proteins are important within the cell. They're the main, again, constituent of the cell. If you were to take a cell and dry it down, remove all the water from it, then 2/3 of the remaining material or dry weight of the cell is made up of proteins. So proteins have a lot of different roles within the cell. A lot of times people think of them as only being used to build muscle. That's not true. So protein has many roles, and here are just a few of them. So protein functions. Each protein will have its own specific role, and we'll hear lots about them later. Some proteins are found in the cell membrane. Other proteins are enzymes, and they can be found in a variety of locations. You'll hear a lot more about enzymes and how they work in a biochemistry class. We'll talk about enzymes in this course, but not really focus on the kinetics of them. Other proteins function as cell signaling molecules, perhaps as receptors, which you would have heard about in an introductory biology course. And then there are other proteins yet that have structural roles. So they don't catalyze reaction like an enzyme. They have some sort of three-dimensional role. And one thing that we'll hear a lot about in this course is the cytoskeleton. So that's made up of several different types of proteins. Another topic for this course is to talk about motility. So some proteins are used in adhesion and allow cells to move or crawl. And then there also are proteins that are involved in transport of materials within the cell. And finally, in this course, we also talk about how proteins themselves are transported throughout the cell. Page 122 in your textbook has a nice panel which is describing a variety of different protein functions. This is just to give you an idea of the roles. This is not a complete list, but just a way to give you an idea of the diversity of proteins out there. And again, start to understand why we're so interested in them for functioning of a cell. To get back to the key parts of the structure of protein, though. We're going to talk about amino acids in more detail now. Basically, each protein, again, is made up of a polymer of amino acids. Typically, this is going to be hundreds of amino acids that are bonded together in one long covalent chain to make up a single protein. And protein shape is determined by which amino acids are present. And that's a lot of what we'll talk about today. Hopefully this is review for you from an introductory biology course or other science course that you've taken. And if so, take this as an opportunity to make sure that you're picking up on all of the details for this. Protein shape, again, is determined by which amino acids are in a particular protein. Okay, so let's get back to talk about the structure here of an amino acid. This is shown on page 74 of your textbook. And basically what we see here is the amino acid structure. So this is made up of several different atoms. And basically the way I like to think about this is how it's built together with carbon at the center. So there's a central carbon, and then there's 4 things that hang off of carbon. As you know from other courses, carbon can form 4 bonds stably with other atoms. And so basically from that central carbon, on one side we have the functional group called an amino group, which is NH3 plus here. This particular amino acid is shown at pH 7, and this is when both the amino group as well as the other major functional group here, the carboxyl group, are each ionized. So again, what is shown here is at pH 7, the amino group has a positive charge and has 1 additional hydrogen, and the carboxyl group has lost a hydrogen. So amino group and the carboxyl group are ionized. This is what an amino acid structure would be within a cell. Sometimes you'll see drawings in textbooks that show the non-ionized form, in which there is the amino group is NH2, and the carboxyl group would be COOH. All right, so now we've talked about the 2 key functional groups, amino group on one side of the central carbon, carboxyl on the other side. Let's talk about this last group down here, which is the R group. This is the variable group. And this is what provides difference from one amino acid to another. Before we talk about that in more detail, I'm just going to point out the very last part of the amino acid, which is a hydrogen, just plain old hydrogen up here. And then basically what that means is that this core part, or backbone as it's called, is made up of the amino group, hydrogen, this carbon, and then a carboxyl group. And again, that's the backbone of the amino acid, and we're going to hear more about that in terms of protein shape in a future lecture. So let's talk a little bit more about the R group. Again, that's variable. So the R group in cells, there are 20 different possible R groups. And that's what's common in cells. In a test tube or in a laboratory setting, we could make different amino acids. They're just not likely to be found within cells. So we will focus on these 20 different R groups. You do not need to memorize their structures. However, you should be able to assign which category that they are in. And you should be able to read a chart that has that information. So let's talk about a couple examples here. So the most simple R group is a hydrogen. And so if there's a hydrogen that is in this position here in the R group, then that means that that particular amino acid is called glycine. Again, you don't need to memorize this. I'm just giving you an example. Another pretty simple R group is a methyl group, CH3. And in this case, then the amino acid's name is alanine, or alanine. Now we're going to talk some more about how we take a variety of amino acids and have them in a chain to make a polypeptide. So moving on to this next figure. This, by the way, is figure 4-2 in your textbook. Now we see that there are several amino acids which are bonded together to make a small protein or polypeptide chain. And so what I'd like you guys to do is take a minute to look at this figure and determine how many amino acids are shown here. So pause the video now and sort out what that would be. Okay, so if you're going to figure out how many amino acids are shown, then we need to use our structure of the amino acids. So if we look here, notice that there's an amino group, central carbon, carboxyl group, and it is bonded covalently through a peptide bond to another amino acid. Basically, each of these gray boxes is a single amino acid. And then you can see that there's an R group that's hanging off, and in this drawing they hang off alternatively on different sides. And so basically what that means here is the answer is there's 1, 2, 3, 4 amino acids in this polypeptide chain. So this is really important to be able to sort out how the figures are used, and so I'd really encourage you to spend time looking at key figures and especially look at the color coding. These textbook art authors basically have spent a lot of time color coding things, and so that can be a really useful clue to sort out what is happening. So let's look at some other aspects of the structure here. Notice that on one side of the amino acid chain, or the polypeptide chain, we have what's labeled the amino terminus, or N-terminus. This is the side which has the amino group protruding, and notice there's a plus charge here. On the other side of this polypeptide chain, then there's a carboxyl group, which is protruding and has nothing bonded to it. This is called the C-terminus, or carboxyl terminus, and that's not a coincidence. C is for carboxyl and is referring to the nitrogen in the amino group. And so this is a way that we orient the strand of the polypeptide chain. So be aware that you will be able to, you should be able to label the N-terminus and the C-terminus. And if we talk about both ends, then we call it termini. Whereas terminus is one of the ends. And it can be specified as either the N-terminus or C-terminus. Again, the peptide bonds are crucial. So peptide bonds, this is a covalent bond between two adjacent amino acids in a polypeptide chain. And a peptide bond is C double bond O to the N. The peptide bond itself refers to this bond right here. And we also include within, whenever we write out what a peptide bond is, the C that's double bonded to an oxygen, because that provides information about the context. The C double bond O originally came from the carboxyl group on one amino acid here. And then the nitrogen here is part of the amino group on the next amino acid. As a reminder, or just kind of a question, do you guys remember what enzyme produces peptide bonds in particular? So what enzyme is producing proteins? So pause the video, think about what that is. And the answer is the ribosome. So ribosome, this is the protein producing factory in the cell. More specifically, this is an enzyme. All right, and let's talk a little bit more about the R groups. So again, we've talked about the backbone. Notice that this polypeptide backbone here is shared, and so there's no differences across the amino acids as far as what's made up of that backbone. We see a lot of alternating nitrogens and carbons, and then what makes the proteins unique and leads to their specific shape and function is which R groups are present and in what order along this long polypeptide chain. So basically, let's take a look at those. So you've got 4 different R groups, and so I'd like you guys to think about what properties that you see and try to assign them to different categories. So basically, these are the categories for how we classify amino acids. There's 4 groups that your textbook uses. One is that they have a negative charge. It's a full negative charge. Other textbooks, including the one we use for introductory biology, would call this the acidic group. Then the second category is positive charge, which would also be known as basic. A third category is to have polar groups. This is uncharged. They may have partial charges, but they don't have a full positive or negative. And then finally, the fourth category is nonpolar. So I'd like you guys to think about for the amino acid side chains which are shown. So how would you classify each of those amino acids? For methionine, here we have the R group. And so which of these four categories would you classify it, and do that the same for each of these other R groups. So pause the video now and think about how you could assign those. Okay, let's talk about the side chains again. So let's start with what I think is one of the easier ones, which is to look for these charges, full negative or full positive. So if you look at each of these amino acids, the R group that has a full charge is here aspartic acid, that it has this negative charge. And so we would classify aspartic acid as acidic, or having a negative charge. So let's write that here. And your book's going to stay consistent with this color coding, so blue ends up being acidic. Then let's take a look at the green R groups. And so basically what you see for each of those is that we've got a lot of carbons and hydrogens. And so think about what that would be as far as a charge. And basically those end up being the nonpolar groups. So methionine is classified as a nonpolar amino acid due to its R group, and likewise leucine is also classified as a nonpolar amino acid due to its R groups. And then finally, let's check out what tyrosine is. That's what's shown in yellow over here. So tyrosine has this R group. And so this one's a little bit curious because it has a ring, which that's lots of carbons and hydrogens, but it also has an OH. And so basically what you need to know is that the OH group wins out as far as the properties. And so an OH group is a polar group. And so overall that means as we classify these and as they're tested in a laboratory, then this would be a polar group. So anytime you see an OH, know that that would be a polar group. So polar groups, again, would have an OH. Nonpolar would be lots of carbon and hydrogen. And they could be in a chain or a ring structure. And again, these are going to show up as green. Polar is going to show up as yellow. And then, so far we've only seen one of them, which is the acidic, and so that's blue. And the basic is not shown in this particular figure. One more thing is to consider how these would interact with water. So in a cell, cells are primarily made up of water. About 3/4 of the cell is water. And so basically we're always interested in how they interact with that. And so basically these first three categories, those are all considered hydrophilic. And nonpolar is hydrophobic, so water-fearing. And basically, I will use those terms interchangeably, polar and hydrophilic, and hydrophobic and nonpolar. And going on to our last slide of this video. So now I want to show you a chart which has a list of the different amino acids. This has a nice review on pages 74 to 75 of your textbook, so I'd encourage you to look at those. This has a list, again, of 20 amino acids that are found within cells. So basically, amino acid names are all along here, and then they've got them color-coded here, and now we can see that the positively charged amino acids are going to be shown in pink. And again, you don't need to memorize that, but that can be useful as you're looking at figures to get more out of them, is to know what the color coding refers to. It's usually not coincidental. Again, you don't need to memorize these, but you do need to know how to interpret those. And this by the way, is also specifically figure 4-3. And so one of the things that we see here is not just that it's a list of the different amino acids, but they are split into their categories. So we also see their chemical properties here, as far as negatively charged, positively charged, uncharged, polar, or nonpolar. I typically like to use the 3-letter abbreviations, just FYI, but sometimes figures will use this single-letter abbreviation. This is the full name of each amino acid, and then again their properties. And why we're so interested in the chemical properties is that the chemical properties of each amino acid and the particular constituents of the whole amino acid chain, which again can be hundreds of amino acids, those R groups, the side chains, and their chemical properties are going to determine the shape of the protein. And so basically, this will form the three-dimensional structure of the protein. This gives it the protein shape. This occurs through protein folding, and that particular shape of the protein determines its function. Okay, so a few notes about that. So basically, why we care so much about chemical properties of each amino acid. This determines how it interacts with other amino acids in that protein. And the most important thing for this course is to understand that those interactions will produce the shape of the protein. So basically, through protein folding, this produces the three-dimensional shape of that specific protein. And form follows function, so the shape of a protein determines what its function would be, whether it is an enzyme or it can, for example, bond with a certain ligand or receptor, and so on. Finally, one other reason why we're so interested in keeping track of these categories is that if there are mutations in the DNA spelling, which then would potentially change the amino acid encoded, we need to know if it stays within the same category, say if negative charge to negative charge, change happens in a certain position, and how that might impact the shape of the protein. So mutations. So basically, if mutations occur, it's important to know if the chemical properties have changed for an amino acid in a certain position. And again, you're going to hear a lot about mutations if and when you take a genetics course, but we talk a lot about mutations in this course. Mutations are the ways that we can determine what happens when something is tweaked. And so we will be talking about those mutations, and again, the properties of the amino acids can be a key part of predicting and understanding the roles of a given amino acid.